Okay, welcome back. So we are going to talk about gel electrophoresis now, specifically of nucleic acids, and then go into a little bit of how proteins work and how we can separate those. So when we're talking about DNA, this primarily separates the DNA molecules by size, which means we can use it to determine how pure the samples are, or are there multiple sizes of DNA in that sample, and then we separate them out, or can play with them, or manipulate them, or do all sorts of things with them. So let's talk a little bit about the setup. This is one way to do it. We have a gel in the middle. The gel is surrounded by buffer, especially on the top and the bottom, and then in between this little orange slab is the gel, and it's surrounded by buffer on either side. We put the sample we want to separate in these little wells at the top. So it kind of looks like the top of a castle tower. We put the DNA in the bottom part of each one of those wells, and then from that well, as the material and sample is moving through, it's going to separate out, and we call that a lane, everything that comes from the same well. Okay, so we've got buffer, we've got a negative electrode that hooks onto one side of the gel, and a positive electrode that hooks onto the other side of the gel. It doesn't actually touch the gel, it just touches the buffer that the gel is connected to. And since DNA has a negative charge with the phosphate group in the backbone, it has a negative charge, which means all of the DNA from this well is going to move towards the positive electrode, because those phosphate groups in the backbone are going to move towards the positive electrode, and they will separate out by size. So that's what this is saying. After it's all run and it's stained, now you can say, okay, well, this one has three different sizes of DNA in this one well, and well in lane number two. Well three, as they move through that lane, it only gave two different sizes of DNA. And then remember, we always have at least one lane, and at least the first lane that has the ladder with known sizes in it. So just remember, DNA migrates by primarily size, and the smaller sizes move through the gel faster. That's a really important thing to remember. It may not be intuitive, but the smaller ones move faster. They're able to kind of wiggle their way through the gel better than the longer pieces are. Okay, so this is what one looks like in real life. A gel, inside of a gel box, there's buffer. This is a little reservoir or tank, and then there's one over here too, and you cover the gel with the buffer. And then this little pipette is squirting a little bit of volume of DNA into that well. And then after we put the lid to the gel box on, you see the red electrode is going to be the positive one, is going to be at the bottom of that box, which means the negatively charged DNA is going to move down through the gel and towards that red electrode. Okay, so once again, we always have at least one ladder, one lane with ladder in it. That's at least lane number one. And we can't see DNA to begin with. I should go back to this one and say we can't see DNA. We do add a blue dye in there. One, so we can kind of see where our sample is in the well, and two, as that the dye also helps the DNA, it sticks to the DNA, makes it heavier, so it doesn't just float out of the lane or out of the well and just we can't see it. So as the electrode is attracting the DNA, that dye can separate a little bit, and it helps you know when the gel needs to be turned off, when the power supply needs to be turned off. Then we need to stain the gel. There's a few ways to do it. One way is with ethidium bromide, which can cause cancer, but the cool thing with this one is it does fluoresce when there's UV light exposed on that gel. So when the UV light is on, you will be able to see these bands in the gel, and when the UV light is off, it'll just look like a piece of agarose. Okay, and then we wanted to use this band to do something, the DNA in this band to do something else. We can actually just cut that DNA band straight out of the gel with an X-Acto blade, put it in a tube, dissolve out the agarose, and then and keep the DNA afterwards. So this is how ethidium bromide works. It's a relatively small chemical, but it does slide in between the nucleotides in the double helix. They call that process intercalate. Um, and that's what makes sure the DNA glows and the gel doesn't really glow. Okay, so some rules. of electrophoresis, again, we have always at least one lane with ladder in it. In this case, there's one on the far left, lane number one, and one on the far right. Again, the smaller pieces move faster, and we always position the gel so that the wells where the DNA started running from, that's always at the top. Okay, which means the smallest fragments are always going to be at the bottom. All right, so if couple of scenarios, if we have two bands that are about the same intensity, then we can say that they have roughly equal amounts of DNA in both of those lanes. However, we cannot compare intensity of a band up here with the intensity of a band at a different vertical spot on the gel, because the longer the DNA fragment, the more of that ethidium bromide is going to stick in the helix, the brighter the signal you're going to get, even though uh, there may actually be more copies down lower. You just have shorter fragments that's absorbing less ethidium bromide, and that's going to make things hard to interpret if you're trying to compare this band towards the top with this fainter band that's towards the bottom. You just can't say there's more DNA up here than there is down here. You can say this is a brighter band, but you can't say anything about more or less DNA. If the bands are at the same spot or close to the same spot, you can say there's less DNA here in um, the second part of lane 9 than there is in this lane. You can say that. You just can't compare and say there's more DNA in one band than another if they're not at a similar vertical spot. Okay, so that's it for DNA electrophoresis. Just a quick reminder, we can run DNA gels using either agarose as the gel material or polyacrylamide as the gel material. Polyacrylamide gives you better resolution than agarose does, but agarose is better, cheaper, and faster to use. It's also not it won't cause any harm. Polyacrylamide is actually dangerous until it's polymerized, until it's solid. As a powder, it can be dangerous. So they used to use the polyacrylamide gels when they were doing sequencing, but now we have the sequencing machine do that instead. Okay, so if we're separating proteins, that's a little bit different. We can only use polyacrylamide to separate out proteins. The trick with proteins is that some of the amino acids in the protein can be positively charged or negatively charged or have no charge. So that's very different from DNA. So we use a little trick to make every protein have a consistent negative charge. We'll talk more about that in a second. And then proteins also have some crazy structure. They can have primary or secondary or tertiary or even quaternary structures. And so we add another chemical that breaks apart all of those structures. So it's just the primary sequence of the amino acids that are going through the gel. Okay, so when we use polyacrylamide to perform gel electrophoresis on a protein, we call it a PAGE gel. That means in this case, the proteins are not modified. We haven't chemically changed their charge or their shape or anything. However, if we do change the size and shape, the chemical that causes that is called sodium dodecyl sulfate, or SDS. And then we add those three letters onto the page that we already talked about. So this is going to be SDS-PAGE, or sodium dodecyl sulfate polyacrylamide gel electrophoresis. That gives the protein a consistent negative charge and breaks apart the secondary, tertiary, and quaternary structures. So the way it does that is in SDS, it's basically a kind of a fatty chain with a negative charge. So this chemical sticks to the peptide, forces it to kind of unfold itself and become linear, and then there is a negative charge at one end of that SDS molecule. So now you have a consistent negative charge. Smaller proteins are going to have fewer negative charges because there's fewer SDS bound. Larger peptides or proteins are going to have a more negative charge because they're going to have space to have than more SDS molecules bound to it. The second trick is we add this beta mercaptoethanol, and they abbreviate that as BME. That is what destroys disulfide bonds and helps to denature the protein. So that also helps make everything linear and easier to move through the gel. So then if we load this, this is the same sort of setup. We've got wells at the top, runs the sample down a lane. We've got buffer on each side. We're going to have electrodes. And again, we're going to have the positive electrode at the bottom because these proteins are now going to have a consistent negative charge after that SDS chemical is added. And then they'll separate primarily by charge or by length, number of amino acids. So originally we have two proteins. One of them actually has quaternary structure, so two different peptides that are covalently bound together by that disulfide bond, and then a third one that's just all by itself. So once we add the beta-mercaptoethanol and the SDS chemicals, then the disulfide bonds break. And now if we were to visualize the top version, uh, the top protein, instead of being one band when we're using SDS gels, it would turn into two bands here. And then protein C would run in a different spot. So that is how gel electrophoresis works in proteins and in nucleic acids. And thanks for watching.